Hello everyone. So we're going to be, so now we know how the solar and the planetary disk formed and how we may have some planets. So now we have a series of presentations about the habitability of the Earth, and then you will hear tomorrow, I think, about Mars and some exoplanets. So indeed, the Earth we all live on, so there is a lot of life on Earth. And so far, it's, and to the best of our knowledge, it's the only planet that hosts life with this constraint that that's the form of life as we know it. It doesn't mean that that's the only forms of life, so we would need to know what we're looking for if we're looking for life on some other planets. And it is actually very unlikely that Earth is the only inhabited planet. So before we move on with some facts for the Earth, I would like us to go together through this slide that's been proposed by the NASA in terms of the criteria that make a planet habitable. So whether a planet is habitable, or which means also can host life, doesn't mean that it's inhabited. It means there could be life there. So that's an important point. So this depends on the complex network of interactions among the planet, other planets in the same stellar system, and the star they orbit around. So the standard definition for a habitable planet is one that can sustain life for a significant period of time. That's an important aspect. It's based on our experience in the solar system. And as far as we know, so life requires water, so liquid water. It requires energy and it requires nutrients. And again, it is as we know it today on most of the Earth. We'll see that there can be some subtle changes depending on where we're looking on the planet. So the habitable zone is the region where around the star where the planet can receive the perfect amount of heat to maintain this liquid water on the surface, and this is the case current days for the Earth. However, as researchers discover unexpected environments that can sustain life, the requirements for habitability on exoplanets are being redefined. So, and in particular, as we are learning that potential habitable zone, we know that all the planets may not host life. So an example, for instance, in the habitable zone is a planet. The example is that of a planet that would be tidally locked and that would show always the same face to the star. So then, in that case, this would lead to an extremely hot face of side of the planet and an extremely cold one, and that would potentially not work. And there are some other considerations that are broad, but I took those that really apply to the Earth, because that's really the basement that paved the way to define the search for life elsewhere. So I thought that it would be good, as we are defining habitability on Earth, is to spend quite some time actually to know what are we talking when we talk about life on Earth and the kind of habitats that are inhabited on Earth. It's not exactly a trivial thing, and that's not necessarily something you all probably not all think about. So here we do have an inventory of the number of cells on Earth in the different environments, in the different habitats. So currently the inventory, we know that on the Earth we have about 10 to the 30 cells inhabiting the Earth. So, and this is mostly archaeal and bacterial cells. And most of them are actually not on the surface, and the eukaryote cells, so I will talk about it in the next slides, but it's orders of magnitude below in terms of cell numbers. So most of the life in terms of, I would say, if I may say, individuals. So those individuals, they live in the deep oceanic subsurface for almost half of them. The other half is in the deep continental subsurface. Then we go with one order of magnitude less in the upper oceanic sediments, a little less in the ocean, and we have also a bit in the soils. And then, so we're like in the orders of the 10 to the 29 cells in those habitats. Then when we go toward the surface, like groundwater, phylosphere, the cattle, termites, pigs, humans, seawater layer, atmospheres, etc. So we're decreasing by 1,000, by 10,000, by 1,000,000 in terms of cell numbers. As we do, those cells, most of them they don't live as individual cells. Most of them live in biofilms, so in colonies, and so they help each other with different metabolic activities. So I think that's important when we try to look for life elsewhere and to define the habitability of the Earth and where it is, how to the parameters that have to be taken into account later. So, since I've started to talk about archaea and bacteria, so we'll go to the next slide, and just as a quick reminder, for those of you who might not be biologists, so the life is divided in 3 large domains, with the bacteria, with the archaea, with the eukaryotic cells, and we are here among the chordates, and we are at the top of one of these little branch. So this tree of life here is not perfect, is not really exact. A schematic one that would be more appropriate is this one with the different way the 3 domains are rooted. So we do have the domain of the archaea that is branched with the shared domain between archaea and eukaryota, and they have what we call the LUCA in the past. That is the least universal common ancestor. And one important point is that it might not be a single organism. So it has a number of common properties to all life on Earth, but even if we looked in the geological record, we might not find, it is very unlikely actually that we find one organism that has all the properties of the LUCA. That's how life was at this point. So I described a bit the number of cells, so the individuals that are on our planet with us. Now it may change when we start to consider the size of the things and the life we may look at. So here this is a scale, so we go from 0.1 nanometer to the atoms to the humans, and how we can, the observation tools that we may use. So the large size above tenths of a millimeter, we can see them with our eyes. Then if we go to a little less than a micron, we can use the light microscope, and then we have to go with electron microscope. So most of the cells I've just talked about, they are located in this range, so around the micrometer size. So we're talking about a large number of cells, but they are very small, and so they are very difficult actually to see. So that they require it's difficult to see them with the light microscope unless we do have some specific staining, or we have to use the electron microscope. And when we talk about the eukaryote cells, so you see here an example of an animal cell, of a cell plant, of a human egg, so those are much larger. So that brings me actually to the next slide. But before I move on, so just a quick information about the difference between bacteria and archaea. So they have tens of difference. But basically, if you want a general idea, so they are very different mostly in their membrane and in their cell walls. So the bacteria membrane is made of ester-linked lipping with D-glycerol. They have basically a straight chain. Whereas in archaea, this is ether-linked lipids with L-glycerol and this branch chain. So for the cell walls, they have also different cell walls, but I'm not sure we really need to go into details. We can talk about it later if you're interested. So given the different sizes of the different in the 3 domains of lives, So this is now interesting to move on at looking at life, not in terms of cell numbers, but in terms of mass. So here, that's a publication that's convenient in terms of the description of the biomass and its distribution in the different Earth habitats. So what you see here is that the archaea and the bacteria that were most of the cells in terms of cell numbers. So now they are a lot less in terms of mass. So at present day, so most of the mass in terms of the biomass, this is currently in the form of biological cells among plants. So that's about a little less than 500 gigaton of carbon. And the other ones are a lot less. Now if we go to some cells in organisms that we are really interested in, like the animals, which we belong to. So you see first that it's only a very small mass of organic carbon, biological carbon. Most of it is described between the corals, the cnidarians, as well as the fish. And then in the mammals, I find interesting to highlight that we actually, as human beings, we have been really good at colonizing the planet. And we actually represent, in terms of mass of carbon, more than all the other wild mammals. So that's something. So that's how you can see also life on Earth. So the individuals and then the carbon mass. So and it's different, and it's mainly related to the size of the cells. But all of these big cells here, they appeared much later in the evolution as well. So the specific shape of the polygons here, they don't have any specific meaning. It's just for conveniency. Now we can move on and looking at how those that carbon is distributed in the different environments, in the six domains of life. So what we see here is that most of the biomass in plants are in the terrestrial domain. We have quite some amount in the deep subsurface, and we have a little bit in the marine environments. And these distribute very differently now when we look at the relative abundance of these domains in the terrestrial, marine, and subsurface as a fraction of the biomass. So you see that the plants are almost 100% in the terrestrial domain. Then the fungi are also mostly terrestrial. Then we do have the protists that are a little less than half in the terrestrial domain. And as we move on, so we're going to have the animals with most of them in the marine environment. And then when we move on with bacteria and archaea, I think you got the message that they are mainly located in the deep subsurface. So that's really interesting to look at it. Then we can look also at these organisms and this biomass and how they work and how they are related to each other in terms of trophic chains. So where are the consumers and the producers located? So on Earth, right now, so at present day, so in the terrestrial domain is dominated by the producers, while the marine domain is defined mostly with a ratio of 1 to 5 with consumers. So there is a balance between the producers in the terrestrial environment and the consumers into the marine environment. So that's how life is shaped now at present day on Earth. So just a quick information as well. When we talk about the deep subsurface, and in this publication and in many others, so we are defining the subsurface by everything that's below 10 meters below the continental surface. And that's the same also in the subsea floor. So that excludes the soil that really interacts on a very short time scale with the atmosphere. As we mentioned, so most of the biosphere in terms of individuals, so the cell numbers, are located as a function of are located at depth, and so I wanted to show how this distribution, those cells are distributed as a function of depth, but also to give you an idea that we also have cells in the atmospheres, in the clouds. So when we start with the cell numbers at the surface, here this is a graph that, so those two graphs, the left one is for the subsea floor, the right one is for the subcontinental, and then I will talk later on about the clouds. So we do have this distribution, so it's in log of cell concentration as a function of depth in log. So we are here 10 meters and we are here a kilometer below the surface. And what you see is that it's very common that we do have cell numbers on the order of 10 to the a billion of cells per cubic centimeter of rocks or subsurface material when we start from the surface, and then as we go as a function of depth, so there is a logarithmic decay of the cell concentration until we reach the detection limit of cells. So here the limit in terms of cell densities that you see is located around 10 to the 1,000 or 10,000 cells per cubic centimeters. This is not an absolute and a theoretical limit. This is related to our current ability and technological ability to detect cells. If the technology progresses, and it's been the case over the last 2 decades in particular, when I started to look into those data, so we're potentially limited to 10 to the 5 cells per cubic centimeter. So now, after a decade, we reached 10 to the 4, so an order of magnitude. And now, in some of the most detailed studies, they even go to 10 to the 3 or 100, even 100 cells per cubic centimeter. So I want to stress that point. So this is not an absolute limitation. So that's really deeply related to our current ability, technological ability to detect cells. So you see that there are a lot of cells as a function of depth and at least to a kilometer. And most of the studies and the compilations stopped at about a kilometer, a little low here. You see it was a paper in 2012. Here it's 2018. And so as we move on, so the compilations go deeper and deeper. Here it's in the subcontinental, and you see here that they went to 10 to the minus 2.5 cells per cubic centimeter, and even a little lower. It's a little easier in the subcontinental seafloor due to the kind of samples that are handled. So for the subsea floor, so it's often soft materials, and they can be subjected to contamination. Whereas in the subcontinental, so when we reach the depth of a little more than a kilometer, so we're already in the hard rock, and this is often some deep water reservoirs that sometimes have been also isolated from the surface for millions of years, and so it's actually easier to collect the water and to do cell counting in the water rather than having to count the cells into the sediment. So there are some very practical limitations in the way we define the cell concentrations. So, but there is this decrease, and then we don't know exactly whether that's really hit the limit at some point at depth, or there is maybe a curvature and this could extend lower. So and when we try to think in terms of searching for life elsewhere, so that might be actually a little challenging if we want to look at this kind of life as we have it mostly on Earth. So a bit about the cells in the clouds, and that might be also more practical in terms of looking elsewhere than on Earth. So because we do have some an intense water cycle at the surface of the Earth, including in the atmosphere. In the clouds, the more people look at the scientists look at the clouds and observe the cloud and also do microbiology in the clouds, they find cells. here, that's a picture. So it's a really bad example because the authors didn't put any scale bar on this one, but that's how it is. It's probably, I would say, 100 micron or a couple of 100 micron for these green algae cells that were collected in the clouds that hit the top of the Puy de Dôme. There is an observatory there that is dedicated to some experiments and measuring the properties of the clouds. And in particular, so those are green algae, and their genetic characteristics show that they are not local ones, so it's not algae from the top of this volcano. So they potentially actually come from the Atlantic Ocean, and not only from the coast. They potentially are from further away within the Atlantic Ocean. So that's a way to see that even in the atmosphere, there could be actually some life. And when looking at different planets, so that could actually be more convenient than looking at the most abundant one within the deep Earth. So you have less on the top, you have more in the deep, but in terms of conveniency, so I let it to the people doing actually the experiment and the analysis there. They are, of course, so the units, of course, are different because they are measured, the density is measured in terms of number of cells per cubic meter of air. So usually, so the classical range is from 1 to 10 to the 5 cells per cubic meter of air. So that's something that's doable. So basically, we are on a planet where there is life everywhere. Everywhere we've looked for life, we found some. And if we didn't find some, it was most of the time due to a technical or limitation. And when the techniques have improved and we went back to the same places, so we've been able to look for life. I say we, but I've never, that's not the kind of thing I'm doing, but I'm talking generally as a community. So now we'll go to what this, in particular in the subcontinental environments, what does this represent in terms of the volume or the area on Earth that could be inhabited. So we take, so that's I will refer a number of times to this little logo. It's a book that's been published in 2020. I had the privilege to be a co-editor of this book, but it's not the most important. The most important is that it collects the work that has been done by a couple of thousand scientists over 10 years looking at the deep carbon cycle. So whether we're talking about biology, and that goes down to the carbon in the core. So in this book, you will find a lot of things and interconnection between the interior of the planets and the outer parts of the planets. And I will talk about this in a number of slides. So here there's this magic number, 120 degrees C. Who knows what 120 degrees C is? What does this represent?
Limitation of the life.
The limitation of the life. Okay, that's a broad definition. So it's a little more specific than that. So that's the maximum temperature to which an organism, which is a hyperthermophile, has been cultivated in the lab. Okay? So that's something very specific. And because I showed in the last slide that we don't exactly know where is the limitation of life at depths. So for conveniency, that's what we currently take as the limit of life in terms of temperature. It's not absolute, that's what, as of we know it today. And so Cara Magnabosco, so she's taken the different geothermal gradients on the different, so she's made, so that's how she's done some inventory. So she made a little grid of the continents and then assigned a geothermal gradient to each of the little square here. And then with the appropriate geotherm, so she converted that in colors. And the colors represent the maximum depth for life in the continental settings. So you see with the light yellow, so it's limited to less than 3 kilometers, and we move on. And you see that in the places, in the old continental part, in the cratons basically, where the geotherm, so the evolution of the temperature as a function of depth, is the lowest, that's where potentially there could be life down to 20 kilometer depths. So that's never been proven, but theoretically, if we take that limit of life at 120, 22 degrees, that's how deep biosphere could thrive within the continents. So the situation in the oceanic domain is very interesting. It was in the early 2010 within the International Oceanic Drilling Program, which is now the International Oceanic Discovery Program, and that will unfortunately stop at the end of 2024. So the ship that used to do all the drills that are necessary is going to the end of its life, and also due to the cost of energy. So the international community, that's an initiative that is led by the National Science Foundation, so it will end operation at the end of 2024. So there won't be any more call for drill at this point. So, but at the beginning, the first half of the 2010 years, so there was an expedition that has been called, so it was the IODP expedition 370, and the name of this expedition was T limit of life. So scientists drilled in the Nankai trough of the I think it's the Shikoku Island, and they picked up a particular location where the temperature gradient is super high. So with the idea that without drilling too deep, they would quickly reach this 120-22 limit of life. So that's not exactly what happened. So here you have a number of graphs. They're a little crowded, but you have here, so they drilled down to about a little over 1 kilometer, so 1.2 kilometers. They counted the number of cells, active cells. So the field point, that's the cell counts that are above the detection limit. And the empty ones, that's where the counted cells, but that was below detection limit. And that's the same here for the endospore concentration. And there's a correlation with the environmental factor along this core. So there was a fluid all the way down with varying salinity. So starting something that was probably close to the salinity of the ocean, then it decreased, it stayed kind of low, and then it increased again below a kilometer. Then we do have also here the temperature. So, well, we were at the limit of life. And at 120 degrees C, so they also expected to find the limit of life, because there's also a difference between the limit of light found during cultivations in the lab opposed as the maximum temperature in nature. Temperature in nature usually, so the common assumption is that we would not find anything above 70 degrees C. Because at that point, I will talk about it later, it's a bit expensive in terms of energy. But that's not exactly what happened. And you have here the major zone in terms of what's there. So the trench with sediments there, the basin transition, sediments from the lower basin, some hydrothermally altered sediments, etc. And what they found is that that's something that's actually very common when we describe the cells in those environments. That's everywhere, each time there is a discontinuity, then there is a kind of increase in terms of cell numbers. So that's the case here. So you see there is a logarithmic decay of the cell numbers, then it raises again when we reach this discontinuity here, then it decreases again, stay, well, almost, so if they have stopped the drilling at 1,000 meters, so they would say, well, we found the limit of life. But now, so you see it starts again, and it starts again in a zone where there is apparently some hydrothermal activities. Here, there is a décollement, so there isn't so much active cells there, but it triggers a lot of the rays of the endospores at that point. So again, the field point, that's for everything that's above detection limit, and the empty ones they've looked for, but it was below detection limit. The kind of work that is done to do all these cell counting. So here it's a fish, a green staining for the vegetative cells, and for the mostly here we do have a lot of archaea, so that's really difficult to see them. So here that's an example of the sediment or the rocks in which cells were counted. So there are some greenish bright spots, so not my laser pointer, but some other ones, potentially here. But here, just at the top of the arrows, you have little round cells, a little less than a micron, and that's actually the archaea cells that they've been looking for. So that's how they saw that there is indeed some activities. There are cells there, and not only there are cells there, but in terms of metabolic activity, they are actually active. So that is done by taking the cells in the lab, putting them with isotopic labeled nutrients, and then using ion spectroscopy to look how that works. And they found that in this zone, in this particular zone between 100, so below 1,000 meters, and in the range between 100 and 120 degrees C, there is isotopic evidence for hyperthermophile that perform acetate degradation. So they use some organics that are in the sediment and to make their energy. So not only there are some cells to some reasonable concentration, but at that depth, the cells are active. Okay, so that was the T limit of life, but it's no more the T limit of life. So obviously, the temperature limit for life in nature is now beyond the 20 degrees C, and it's an open question. So there's a lot of, you have a bright future on those topics. And in terms of metabolic activity, between 80 degrees C and 80, 90 degrees C, so that's mostly methane. Methane is produced and oxidized in the community that is down there. So I've showed you two examples for the continental setting, the oceanic setting. But now, so here that's a compilation that's been proposed by Merino and collaborators. So the different environments where life has been found, and even in some of those extreme conditions. So you see here that going to very cold location, to deep sea anoxic lakes and brine, we do have the deep sea hydrothermal vents, and there will be a presentation on that particular topic. I think it's on Thursday. So there is the deep sea sediment and trenches, and I will talk about it. Marine and continental subsurface, we've elaborated on that. Soda lake and hypersaline lakes, nuclear contaminated sites as well. All the ophiolites are pretty rich in terms of life down there. Also the acid mine drainage, although it looks very inhospitable, so it has a particular, there are particular forms of life there with communities that are super efficient. Even in the desert and the arid environments, the hyperacidic lakes of the volcanoes, the hot springs, mud volcanoes on the surface, but also in the ocean, some of the shallower hydrothermal vents, and the cold seeps in the mud volcanoes. So basically, in the very soft environments, as we always think of life, of course there is life, but there is also rock-powered life in all the subsurface environments, whether they are mild or pretty extreme. There's been also, so since we know that temperature does not stop at 120 degrees C, there's been a renewed interest in trying to go deeper and deeper. And in those slides, so there's no direct evidence for life, there's no cell counting, there's no cell observation in those particular environments, okay? But I'm going to try to show you how we can also find some other evidence of life. So here, this is another international oceanic discovery program cruise that has been laid in the Isubona Mariana subduction zones. So the number of colleagues have investigated serpentine mud volcanoes. So they go from, so you have here in the subduction zones, you have the subducting slabs, and there are mud volcanoes that arrive to the surface, and they manage to bring some deeper samples. So there's been one here, the South Chamorro mud volcano, and there's another one, so that's been studied in detail by Plumper, and there is another one that is a little northern, that's not on this picture, but that's exactly the same setting, that's been investigated by Debray. So in those mud volcanoes, so you find them at the surface, or when so basically they work on those deep samples. When it goes to the surface, of course, there is always the issue when you look at life if it's been contaminated by the surface biology. So, but looking at those deep samples, these serpentinized diapirs, so they are soft enough that they can bring a lot of different kinds of rocks, and including some other serpentine minerals. So in this setting, so this distinguish the deep forms of serpentine. Its name is antigorite, and that is usually a brown or green. And they found also blue serpentine. And so it was so they looked in detail with high resolution techniques. So you have here the samples. So they look at serpentine, the blue serpentine clasts here. So the size of the sample that is investigated is about 500 microns. And then you go with a deeper resolution with focused ion beam cutting, and then do infrared, high resolution infrared spectroscopy or some chemical mappings. And you see they end up with identifying a lot of characteristic spectroscopic line that are characteristic of organics. But at that time, so in 2027, so Plumper showed, well, there are a lot of serpentine clasts. They are rich in organic matter, and that organic matter could be potentially from biotic origin. But be careful, on Earth, there is a lot of organic matter that is not necessarily biotic in origin. There is biotic, and there is abiotic organic matter. And it's not always trivial to distinguish between the two kinds. So when I hear sometimes, oh, okay, we're gonna look for life. We're gonna look at organic matter. Oh, okay. Well, how are you gonna discriminate between the biotic and the abiotic organic matter? It's not a trivial question. But on Earth, we know that most of the time it's actually biotic, unless it's very, very deep. Debray and al. On the so they were a little, well, technique have advanced, and they were a little more lucky. So they looked at another mud volcano, so and isolated those blue those blue serpentine that are here in this graph, and I will comment that in a minute. The blue serpentine versus the classical high pressure form of serpentine that is the antigorite, and they didn't look at the organic matter. They actually looked at the carbonates and at the isotopic signature of the carbonates. So that's what you have here in this graph. That's the isotopic signature of the inorganic carbon in the form of carbonate, the heavy carbon, as a function of the total inorganic carbon. And here what you see on this graph is that the normal, I would say the normal antigorite, so the abiotic, typical petrology stuff, that's the things that are happening geologically in the subduction zone. They have a very different signature than the one here with the blue serpentine, the carbonates that are within the blue serpentinites. And that train here, pointing to very light carbon, is actually specific from a biological activity. So the biological reaction, they don't like the heavy carbon. Kinetically, it's not favorable. So they really prefer the light carbon. And the way they could interpret it with some other observations that I don't necessarily report here is that this carbonate light signature is consistent with anaerobic microbial consortia. So it's not only one kind of microorganism, it's a series of microorganisms, it's a consortium. And it's a syntrophic methane oxidation that's coupled with sulfate reduction. So they found actually evidence for deep life in that particular context, to conditions of 20 kilometer depth and potentially more, not looking at the organic matter, not trying to find as hard as they could life into some rocks, but using the isotopic signature. So that's the kind of thing. So potentially, so here there is the 122 degrees C isotherm that's reported in that context of the subduction zone that is already pretty deep in this setting, but maybe in some of those deeper samples, there could be also traces of life that is recorded in the carbonate, in the isotopic signature of the carbonate. So if I want to summarize before I move on with some other geological consideration, so basically, so I reported here the kind of geotherm for, general geological, the geotherm here, this one is a little steeper in ice. So you see that at the surface or subsurface of the Earth, we are clearly within the liquid water, and that continues at depths. It's also when we go into more physical information here, that's the phase diagram of water with the two forms of life. There is a high density water and a low density water. Low density, so that's how the water molecules are organized. You have the low density water in blue, the high density in green. So the life is mostly in the low density water regime. And now, so that's here a graph that represents the limits, the limits of life as we currently know it in terms of the main parameters, pressure. So that's this blue, this line here, the salinity, the temperature, and the pH, and the slight difference between the culture study, that's a little deeper blue here, and the environmental-based study here. So there's a little bit of difference, of course, as you know, in temperature, but there's also the same for pH, the same for so high and low, low and high pH. For pressure, we don't know necessarily very well. But here for the salty things as well. And that actually corresponds also to what we know in terms of the water that's superimposed with the conditions for the chemistry of the Earth, the chemistry of the water on the Earth. Now, if we want to go, I will go a step further with and I will switch to geology. If we try to interface what I've just said with the minimum requirements for life, and what the Earth actually can provide to life. So liquid water, I won't necessarily talk too much about it because that's really the thing that everyone talks about. So I don't think there's really a point to spend some time on this. One important aspect for life in terms of requirement is that it has enough of the element building, the building blocks and some other trace elements that are required for the metabolic activities. So the basic element is that life requires carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. I will talk about most of them, but the phosphorus cycle on Earth, we don't know it particularly well, and there's no published phosphorus cycle at this point. There is a big issue with how to solubilize the phosphorus. It's not a trivial thing. I will talk about energy, about the conditions for which there is most of the time life, is that when we have water, when we have enough of the elements, and when we have chemical disequilibrium, and each time there is electron that can be transferred, there is life. And then the requirements for life, it's also stability over time. although the conditions at the surface of the Earth have changed through geological times. So I will go with how the Earth provides what we call the life essential volatile elements. So here you do have a distribution of the carbon in the different Earth reservoirs. So we have a reference of the bulk silicate Earth here with 1.4 10 to the 9 gigaton of carbon. And then as we move toward the surface, so we have less in the inner Earth, and then there is orders of magnitude when we go to the atmospheric, what we call the exogenic envelopes of the Earth. So there is enough carbon on Earth, provided a little thing is that currently most of the Earth, most of the carbon, is located and stored in the core of the Earth. So we don't have the exact carbon budget in the Earth. So it could range. The concentration could could be from 0.1 weight % of carbon in the outer core to 1 weight %. There could be, in that case, also some carbon stored in the inner core. But there's really a really huge reservoir of carbon within the Earth. The question is it available to the surface, and can it replenish the surface to make carbon available for life? To do that, I will, in the next slide, compare the distribution of these life essential volatile elements in the different terrestrial and chondritic reservoirs. Here you do have, it's represented in terms of a ratio of carbon over nitrogen, carbon over hydrogen, carbon over sulfur, and I added here, although there's not the corresponding graph, carbon over oxygen. When we try to do this work for the interior of the Earth, it's very difficult to go with the absolute numbers. So we tend to work with ratios or with the isotopes, because the inventory is very difficult to make. We have to make comparison with the different meteorites, but how those meteorites actually correspond to the exact composition of the Earth, we're never quite sure about this. Well, we have some good ideas. So you have here all these ratios, and I've compared those ratios with what we have in cells. So here you do have that in mole %, so the relative amount. So we do have more hydrogen than carbon, a little less than oxygen, and a little less than nitrogen. It's a little different for budding yeast. Most of the data are actually available for the human body, but the ratios are not changing that much, and they correspond to those kind of ratios here in weight %. So how does that compare? So here, when we do have a higher ratio, that means, for instance, that for nitrogen, so we have a high ratio for in cells. We do have high ratios here in the cores, means that it's this envelope is depleted. So basically, if we look at, so you have here the bulk silicate Earth, you have the core, you have the mantle, you have the exogenic envelope, and the scientists usually try to compare with the different kinds of meteorites. So what we see is that for carbon, so the bulk silicate Earth is slightly depleted in nitrogen compared to the carbonaceous chondrites that are supposed to be really important in the building blocks of life. For sulfur, it's about the same, and that's the opposite for hydrogen. So you see we go from one side to the other. So what we see, and then we can compare what's happening in the cells compared to the bulk silicate Earth or to the exogenic envelopes. So you see that in living cells, so the carbon-nitrogen ratio is 4 to 10. So it's about the same, or within the same order of magnitude as the exogenic reservoir. For the carbon over hydrogen, it's a little different. So we have less hydrogen here than in the envelopes, and for the carbon over sulfur, it's the same order of magnitude. Also, that can be disputed, and there's no point going over that. So basically, there is enough. This ratio are globally consistent in life and in the Earth, and in the Earth envelopes, at least the exogenic ones. But there's one thing that is important, is that when we go back to the history of the Earth and how the way the Earth formed, and Manuel started to touch upon that this morning. This is that when the Earth first created, so it was hot. There was a magma ocean that formed, and this proto-Earth, Manuel said this morning, so it was pretty dry actually. And if we take that and nothing else happened, probably it is very unlikely that we could have had life on Earth. And there wouldn't be any exogenic envelope that have the suitable concentration that is more or less in accordance to what we have in life. To do that, there are lots, so it's been solved by isotopic measurements on rocks. It's been solved partly, at least, by doing a large number of experiments. And basically, I will make it short, so the only way to do it is to add to the Earth, after the core was formed, is to add it another planetary body, probably that had to be differentiated, quite of a large size, that had a mantle, that was carbon-rich, and that had the suitable carbon over sulfur and carbon over nitrogen ratio, to add those life-essential volatile elements to the mantle that could be later degassed. So that's how it's been done here. And potentially, so that's one of the major events. This large event here, I think that you've heard about it, it's basically the moon-forming impact that happened after the core of the Earth was formed. That was potentially not enough, so it probably required what we call the late veneer. So small, water-rich, carbon-rich particles that hit the surface of the Earth. That's the only way to do it. And the right size to do that, and I will go with another slide. We do have some constraint on the size of the embryo that hit the Earth to make this. It was probably between 1.1 and 0.2 Earth mass. This has been confirmed. So here, so I said that a lot of this has been constrained by experiment, but that's not enough. So here. This study by Grewal and collaborators, so here that's the same scenario as the one that I just proposed, but they had to compile and to use data science to look at the silicon, the sulfur content in the core that's compared with the bulk sulfur in the Earth, and the partitioning between the iron, so the metal alloys and the silicates. To look also at the bulk carbon as a function of the bulk sulfur and another partitioning. Here you do have, so that's the entire set that's consistent with what we know that could happen from the experiments. So you had to change the ratio, you have to change the pressure temperature conditions, the oxygen fugacity, a lot of parameters. And here that's not only a compilation of those data, it also uses data science to produce that, and that's a comparison here with a number of meteorites. So they managed to show that indeed that's something that could be suitable with some of the carbonaceous chondrite, but not all of them. And then from that, they could also assess the size of the impactor, again by looking at those ratios and at the potential carbon concentration. And so that leads to this trend here. And when you compare some known body like Vesta, the Moon, or Mars, there's only something that's about the size of Mars that is suitable. So that confirms also what we knew before and brings some other constraints. So that's how some of those light living elements that were bring to the Earth, it's to this giant impact. The name of the impactor was TIA. So then I will go to another aspect, that's the issue of the energy. So here I just show an important point. That's that's a particular metabolism that's been investigating here. That's the sulfur reduction here. So in terms of femtomoles per cells and per day, so going from a small amount to 0.1, and as a function of depth in energy limited system. So you see that here, so it's really decreasing very quickly as a function of depth. I took this example here to show you that under energy limiting conditions, so a cell basically would need approximately 1 to the minus 70 kilojoule per day. So that corresponds here, just to give you an example, if like this cell would respire 1 mole of sulfate every day, and that for 30 years. So that's a kind of, so it's very, very little. So never forget that when here in this deep part of the earth, so the cells, they have some activity, but they don't necessarily have a very intense activity. So some of the cell consortia, they have replication time of a couple of days, but that could go to 100,000 of years. So it's not one cell, but it's the cell community. So it's very little. So that's probably as little as it's been described in the literature. When we go to hyperthermophilic microorganisms, so such as those that I've described in the T-limit samples, so they need actually more energy because temperature induces a lot strong damages to the cell, and in particular to the proteins, and so the cells need some energy to fix or to repair the damages. So in that case, they need 1,000 more energy per cell and per day. As a comparison, we require a lot of energy. So that's basically what I deduced from our daily diary, recommended diary. And that corresponds to an energy, to a power of about 10 to the 15 to 10 to the minus 6 watt. So it spans a large range. So it spans a large range, but basically, to give you the general idea, so life adapts its space to the energy and the food that is available. The more energy, the more food, the higher the cell density. And then as we go as a function of depth, so the energy tends to decrease, the food tends to decrease, and the cell density tends to decrease. So the cells tend to have a strategy at depth, so avoid dividing if you can. So it's not like, that's from Wikipedia, like not those on the end of 8-year-old kids on a petri dish. And the energy in that case, they take advantage of every electron transferring system that can take. So here that's some of the most energetic one with the oxygenic respiration, but that could be also iron oxidation or iron reduction, depending if it's electron donors or electron acceptors. At depth, so the energy in some settings, there could be also locations where life actually does not necessarily require a lot of energy. That's Everett Schock, who's working at Arizona State University, has dedicated his entire career to calculate the energy of the different metabolic pathways and metabolic reactions and etc. And here it's a very nice example that was published in 2010. So I think it gathered the data for at least 3 or 4 decades of experimental and theoretical work. So they applied all the thermodynamic data that they have gathered to the geological setting of the seafloor hydrothermal system along the mid-oceanic ridges. To make it short, that's this diagram here that represents the cost of methanogenesis at the different so hydrothermal springs. So in the seafloor hydrothermal system. So everything that's above the zero affinity line means that it's exoenergetic, so exothermic as well. It releases energy. So the way they calculate it, because they have to take into account a lot of parameters, so that's how they end up with this representation. And in a few cases, so it requires energy. So you see here that for sites that are well known, that the rainbow, so that's the famous black smoker, but also in many others. So there is a large range of conditions in terms of temperature that at 250 bars, so it's a little below the surface. So such a metabolic reaction actually does not cost life, energy to life. It releases energy and can feed potentially some other microorganism in the consortia. That's for the one metabolic pathway, but it's also the case for the synthesis of some amino acids. Here I've just selected a couple of two amino acids, so leucine and alanine. So you see here also that in some cases it doesn't work, so it always requires energy, but not systematically. And when we go to the glucose synthesis, that's something that requires energy, definitely. If we go at the synthesis of some nucleobase like the thymine, so that also almost systematically requires energy. So that means that for some functions, the cells would need energy, but that energy could be provided by some other metabolic pathways. So that's a system that can work. My point here is that we often hear that life requires energy. No. Sometimes there are conditions where life does not necessarily require energy. It can, from the environment, produce its own energy. So that's a sentence, a quotation from Everett Schoch. So many autotrophs in the hydrothermal systems, biosynthesis act as a free lunch that they are paid to eat. So I think that really summarizes well the thing. So it's been pushed forward by the next generation, Lauro and Amend. So here they compiled data and calculations for terrestrial hot springs, for a shallow sea hydrothermal system, a deep sea hydrothermal system. They calculated the Gibbs free energy of potential catabolic reaction. There's a combination of 19 electron donors, 14 electron acceptors. So they evaluated that for 322 data sets, and the geochemistry, so pressure, temperature, pH, salinity, exact chemical composition, hydrogen, etc., of 30 distinct systems. So that turned into 740 reactions considered. And among those reactions, you see that most of them are exergonic at one or more sites. And that's the case, for instance, of the hygienotrophic acetogenesis, that is a very low energy metabolism, does not require energy. Here that's represented with the reactions, so are the most exergonic on the left and goes toward the less exergonic ones. And you see that for each reaction, depending on the environmental conditions, the energy can span 3 or 4 orders of magnitude. That's the size of each of these little lines. So that's one reaction, but in different conditions. And then when you talk the total, in terms of energy, it spans over 12 orders of magnitude. So the energy in the systems to live may be very different depending on the composition, depending on the metabolic activity that is considered. So it's not a very trivial thing to consider the energy for living systems. Now, if I go to the energy budget, so I think you're very familiar with the energy that is provided to the surface of the Earth. So the solar constant that is divided by 4 because the Earth is running, etc. So, but there's also the internal energy flux from the Earth that is more modest. So you have here schematics of the Earth. It's a compilation of the data plus models. So the average heat flux at the surface of the Earth is 82 milliwatt per square meters. So that goes to that sums up to 42 terawatt at the surface of the Earth, with most of the heat being released along the mid-oceanic ridges and in also subduction zones like here. You have here these really nice Icelandic spots. Hot spots, and we do have some here as well. So the most at the mid-oceanic ridges and the hot spots and the less in the subduction zones. So that's part of it. So basically, I won't detail, just as a reminder. So the water is liquid at the surface of the Earth, not because of the distance of the sun and the Earth, but because of the greenhouse effect of the atmosphere. So we complain a lot about the CO2 level of the atmosphere, but I remind you that if we didn't have the right amount of water in the atmosphere, the right amount of CO2 and a bit of methane as well, so the actual temperature at the surface of the Earth would be minus 18 and not the 15 degrees C as we do have it now. So I won't go. So we are right at the right spot right now. This was not necessarily the case, so I've took that from the National Academy of Science book here that was proposed by James Green. So as a function of time, so we are here, that's where we're going in the future, that's where we were. In terms of so the sun power was a lot less at the beginning. It reaches 100% now, and it will continue increasing as a function. Will increase as a function of time. We are now here. That's where we come from, and we were not necessarily at the very, very, very beginning in the habitable zone. However, Mars was, and Venus was as well, but they evolved differently in the two cases. The stability over time. That's something I want to expand a little more. So. My short message is that if there wasn't plate tectonics, there wouldn't be life on Earth probably over the long period of time. Here, that's the most exhaustive carbon cycle that's been produced by this program, Deep Carbon Observatory. So the amount in the core, the size of the point is that's in log scale, proportional to the log of the amount in the reservoirs. But you see here that there is and you have in blue the fluxes. So you see that you have a deep blue arrow that brings back carbon into the mantle, and you have also a number of blue fluxes that brings carbon dioxide to the atmosphere. The mid-oceanic, on oceanic islands, on the margins, you do have that in the subductions, in the volcanoes as well, mid-oceanic ridges. So there is a very intense exchange and the mantle that buffers the carbon cycle and the CO2 cycle at the surface of the Earth. This can be, so here that's a nice picture. It's better seen when we look at bars diagram. Here you have positive fluxes towards the atmosphere. Here you have negative fluxes, so burying of carbon. So you see that basically what we have now is we have a cycle that is in a steady state. So I'm not saying that it's at equilibrium, but it's at steady state. It's not at steady state all over the planet. If I take the example of the subduction zones, in some cases it may not be balanced, but overall it is balanced. And we do have this anthropogenic contribution to the atmosphere here that is not negligible. You see here from the size of the scale bars, that's a very important component that balances the CO2 concentration on the atmosphere. That's the weathering and the precipitation, of carbonate and organic carbon, and also the silicate weathering. And the silicate weathering basically is due to the distribution of mountain chains at the surface of the Earth. So it's directly linked to the plate tectonics. So here you do have the fluxes. So you see the spans over orders of magnitude, but everything tends to be balanced. But it's not always balanced in terms also of time scales. You do have here the same representation. So we do take the same fluxes, but here there is the response time that is given. So when we're talking about the fluxes from the interior of the Earth to the surface, so we're talking to very long time scale. When we're talking about what's going on in the exogenic reservoirs, it's going more from the year to 1,000 years. So, but basically the long-term volcanic climate is actually controlled right now by the amount of carbon in the exogenic cycle and over the million-year time scales. So this exogenic carbon, so it's also controlled by the outgassing of the Earth's interior, by the volcanism, the metamorphism, or the sequestration of carbon and organic carbon through silicate weathering and photosynthesis. So the response of the silicate weather, the response time for the silicate weathering is on the order of couple of times 100,000 years, meaning that the amount of carbon in the and controls the amount of carbon in the exogenic cycle that controls the climate. So we do have a very strong feedback from the interior of the Earth, and in particular the weathering, to control the climate over the long scale. So the carbon cycle is the one that we know the best. We do have also some drawings for the nitrogen cycles. Nitrogen may really have a lot of spans a large number of oxidation state from the mantle to the exogenic reservoirs. So there's a lot of electron transfers with the cycling nitrogen, but there's a very nice cycle of the nitrogen, and that also strongly relies on subduction zones and on volcanism to be balanced. If we don't have that, it's over, although it doesn't have a very strong implication for the climate. But for life, it would not work like for carbon. We do have here the same for water. So same, the water cycle is also balanced between what comes out at mid-oceanic regions or back-arc basins and what comes in subduction zones. Remember also that we do have one ocean at the surface of the Earth. In the interior of the Earth also, we don't necessarily know exactly how much water we do have, but this could go up to a couple of ocean mass. And in particular, there's one zone that is targeted in the interior of the Earth. That's the transition zone, just so between 410 kilometers and 670 kilometers. And that's a zone where the minerals and the rocks can actually host a lot of water. Like for the carbon cycle, the water cycle is globally balanced at the surface of the Earth and for billion years. However, locally, at some subduction zones, it may not be balanced. So it's a very sweet spot that we have with this system. And that also triggers the control on the redox state of the atmosphere. So that means that on planets where there is plate tectonics, on planets where there isn't, that has a very strong influence and strong control on the composition and obviously on the temperature of the atmosphere. So we are really here at the sweet spot. So Venus, that has exactly the same size, etc., as the Earth, could have become like the Earth, but now, because it doesn't have plate tectonics, it may have had plate tectonics for maybe 500 million years, but that stopped. And now it has a very thick atmosphere, 90 atmospheres, composed mostly of carbon dioxide and temperature of 482 degrees C. Mars is smaller. Mars has lost most of its atmosphere, and we'll talk about it why to some extent. Has very low atmospheric pressure that is also composed mostly of CO2 and has a very low temperature at the surface. So that brings me to the last point here. That is that what controls also the atmosphere, the atmosphere, retaining the atmospheres at the surface of the planet, is also related to the long-term cooling of the planet. Here you do have the Earth dynamo, and it's a picture of simulations that is responsible for the magnetic field at the surface of the Earth that interacts here with the solar magnetic wind. So that's really, if we didn't have this magnetic field, we think that it started on the Earth about 4.5 million years ago. That's the furthest evidence that we do have in the geological record. This is due to the cooling of the Earth and to chemical exchanges between the inner core and the outer core that really helps also the plate tectonics, and probably it really was really important when we thinking about the beginning of the plate tectonics as we know it now. So that's also mandatory to have a plate tectonics, to have the proper atmosphere, and also to have life on Earth. So when we think of on Mars, so the stripping of the carbon and the oxygen at the surface of Mars probably occurred when there was so the magnetic field was not enough, and the stripping of the carbon and oxygen was induced by the strong solar wind that began about 4.1 billion years ago, and this corresponds actually to when the Martian magnetic field stopped. So that it has also a very strong importance, and on the longer term, it also helps the life and prevents cells from mutations. So I will leave you with this summary. So it's a quotation from Anachara and collaborators. Well, the heart of habitability on Earth and potentially elsewhere is in the planetary interior. So on a planet like Earth, well, there is a dynamo where there is plate tectonics, there is recycle, there is the right amount of life essential volatile elements. In other planets that have no dynamics, even if there is some stagnant dynamics, so that systematically leads to a dry surface and there's no life there. So I think that summarizes well the conditions for the habitability of the Earth. Thank you. And I'm happy to take your questions.